As you know, the seminar is held from on the second and fourth Mondays of the month from September through June. And we include speakers with work focused on child health from both UCSF as well as guest speakers. So please let me know via email if you're interested in speaking or would like to suggest a speaker. Looking ahead to our next speaker in 2 weeks, we will be welcoming Dr. Shai Srinivasan from our Division of Pediatric Endocrinology on March 27th. We're still virtual and using Zoom, and we'll ask that everyone stay muted unless you have a question, or we'll open it up to questions at the end. And I think Irfan's going to be a little bit interactive, so please feel free to unmute and respond. Today, we're welcoming Dr. Irfan Kathiriya, an Associate Professor of Anesthesia and Perioperative Care and one of our group of pediatric cardiothoracic anesthesiologists. His research investigates mechanisms of genetic variants and perturbed molecular pathways important in the development of structural heart defects. And the title of his talk today is Patterning the Developing Heart. Patterning the Developing Heart. So thank you so much for joining us, Irfan, and I'll let you take it away.
Awesome. Thank you, Roberta. Thanks so much for this invitation. Let me just pull up my slides. I must say that it's really, Roberta, thank you very much for this invitation, because it's an honor. Since I started my time at UC in the Department of Pediatrics as an intern, and so it's quite a treat to be here to kind of share my work those many moons ago. And I see several familiar faces and many new faces, so thank you all for being here. Today, I wanted to talk about patterning, our efforts to try to understand patterning the developing heart. And so today I'm going to talk really about using genetic tools in the mouse embryo to learn about heart development and congenital heart defects. So as this audience is very familiar, congenital heart defects are leading cause of childhood morbidity and mortality, really the leading non-infectious cause of death in the first year of life. And many congenital heart disease include ventricular septal defects. I think some estimates are up to 50% of all patients with congenital heart disease have some form of ventricular septal defect, and of course some of those are isolated. Up to 20% are isolated. And ventricular septal defects can be hemodynamically significant. They can cause pulmonary overcirculation, heart failure, leading to poor growth, and many of these oftentimes they require some sort of intervention, either by the cath lab or surgery, which is when I'd be involved clinically. And it's thought that VSDs are really the sort of manifestation of abnormal development of the heart, and so and specifically the interventricular septum. So, for example, here's a diagram from the ATOM website. It's a communication from the left ventricle to the right ventricle via this ventricular septal defect. So it's thought to be really abnormal development of the interventricular septum. And so our hope is that by studying normal development, that we can also sort of define what are the disruptions that lead to congenital heart disease, with sort of the long-term sort of goal of trying to improve genetic counseling by sort of broadening the universe of genes that contribute to congenital heart defects or congenital heart disease. And then also, depending on the genes that we, or the pathways that we discover, help with the evaluation of, or leading to the evaluation of other extracardiac abnormalities, as well as sort of futuristic sorts of things of improving tissue engineering, these sort of patterning patches for congenital heart disease, as well as in adult heart disease. So today I was hoping, I'm kind of presenting sort of a work in progress. So please, if you have any questions as we proceed, I'm going to sort of talk about findings and associated techniques for those that are interested. So really the overall question here is, what are the mechanisms that pattern the IVS? And the take-home message today I'm going to talk about a transcription factor, TBX5, which is linked to congenital heart defects. This was sort of shown in the late 90s. And what I'm going to do is I'm going to provide evidence that we define a pathway, a TBX5 dependent pathway of some guidance cues that regulate what's known as a compartment boundary or a lineage boundary for normal ventricular septation. And so here's just kind of an outline of the upcoming results. First, I'm going to talk about a developmental time course of a lineage that contributes to the interventricular septum. I'm going to use genetic lineage tracing using which I'll discuss shortly. Then I'll present some evidence that this lineage that contributes to the interventricular septum, it's essential. We've ablated it, and then it's not only essential for the interventricular septum, but also for the RV and to some degree the LV, and also important for this boundary that separates cells. And then I'm going to present some data that shows that TBX5 regulates this lineage that contributes to the interventricular septum using several techniques, including light sheet microscopy and single-cell RNA-seq. And then I'm going to end with sort of our new findings of how TBX5 regulates new pathways as regulators of the interventricular septum. So the heart is there are at least two. There are three, but here in this slide, I'm going to highlight two heart fields: the first heart field and the second heart field. And each of these heart fields contribute to different parts of the heart. So, for example, in green here, the first heart field, which is the first to migrate from the cardiac crescent, the cardiac mesoderm, and the cardiac crescent to form the heart tube. The first heart field contributes to the left ventricle and the atria. The second heart field, which migrates later and was discovered later, it contributes mainly or largely to the RV and the outflow tract, as well as the atria. And it's thought that basically both heart fields contribute to the interventricular septum, really at this intersection. It includes the interventricular septum, the AV canal, and the interatrial septum. And we've been using genetic tools where specific lineages largely recapitulate many of the aspects of these heart fields. So, for example, the TBX5 lineage, there are progenitors early in gastrulation at the mesoderm stage. As many of you recall, gastrulation is when there's endoderm, mesoderm, and ectoderm. So at the sort of in mesoderm, there are TBX5 positive cells where if you mark them using tamoxifen, so here we're using kind of this sort of scheme, which many of you may be familiar with, where we're using a Cre-Lox sort of scheme, where Cre is a recombinase. It's an enzyme that's fused to the estrogen receptor. So with a ligand, in this case tamoxifen, the Cre recombinase enters the nucleus. It identifies its targets, these LOXP sites, and it causes a sort of excision of the LOXP sites, which removes the stop codon, which then allows the GFP to express. So that's shown here where we're following the GFP. So here you can see in this lineage where we've marked it early, you can see that there are green cells indicating the TBX5 lineage, which contribute to the left ventricle, the left atrium, and the right atrium, as well as to the interventricular septum. Then there is a sort of a converse lineage, the right side, which here is marked by an enhancer of a transcription factor MF2C. The enhancer is called the anterior heart field, just for those that are interested in the details. And this drives a different sort of recombinase. It's called a DRE recombinase. And again, this is fused to the estrogen, modified estrogen receptor. And so here, for example, this DRE recombinase enters the nucleus, and its targets are slightly different. They're specific and different than the LOXP, but the concept is more or less the same. So the ROXP sites, they get excised, the stop codon gets excised, and then the red fluorescent protein can turn on. And so this is how we can visualize both lineages at the same time. So this is sort of simultaneous lineage tracing. And you can see here in this merge photo, you can see green and red, and that both of them contribute to the interventricular septum from each of the sides, and that there's overlap. There are some yellow cells that overlap. And so for much of the remainder of the talk, I'm going to talk about the overlap. this double positive TBX5 MEF2C AHF double positive lineage. But before I go there, one of the sort of concepts I want to talk about is a lineage boundary. So for example, here in this sort of reconstruction, you can see that there's kind of a very sharp boundary of the green cells to the non-green cells. And this is considered a lineage boundary. And this lineage boundary, which you can see here in this cross section, is more or less, it stops at the interventricular septum. And so the cells that I'm going to show in the next slide, these sort of yellow cells, which are going to be represented as red, they're really the overlap of both the green lineage in this slide and the red lineage. And so the double positive represent sort of the boundary front, which is depicted here in this reconstruction. So here in this image, this is a embryonic day 14.5 after the completion of ventricular septation. Here, the green lineage is the same as in the other slide. So this is the TBX5 positive lineage. And then now the red lineage is the superimposed double positive lineage, the TBX5 and MEF2C positive lineage. And I just wanted to go over some of the genetics so that we, just to talk about how we're able to visualize these two lineages at the same time. So as I talked about, there's the Cree reporter. So the Cree binds the LOX-P, excises the stop, and then causes the GFP to turn on. So this is just kind of a standard Cree reporter. And then I mentioned the DRE. This reporter is a little bit different. This is considered a dual or an intersectional reporter. So this is a reporter where there are both LOX-P and ROX-P sites. So this reporter only turns on when both the LOX-P sites are excised and the ROX-P sites are excised. So only in these double positive populations. And so that's what's shown here in red. So you can see that this red population extends from in the IVS from the apex of the heart through the interventricular septum to the base of the heart, through the AV canal and into the interatrial septum. So we think that really that this is a really unique population because it marks this sort of very important site, and we think that it actually marks, as I mentioned, the front of the boundary. We looked earlier in development. I'm just showing you a little bit of data, a flavor of the data. So, so heart development really starts at about embryonic day 7 3/4. So that's in a mouse. That's the cardiac crescent stage where the fields come together that I talked about in the earlier slides. And then the cells kind of migrate towards the midline to form a heart tube, and then they start to have rightward looping. And so this is a stage where you have the heart tube, and it's just before you have the rightward looping. And you can see that these double positive population, these same red cells, which are also here in the interventricular septum, these same red cells are already positioned between the future left ventricle and the future right ventricle. And it forms this really beautifully intricate ring, as you can see here in this short axis view. And this ring sort of expands that after rightward looping in heart development, you can see that this ring sort of expands into a band of cells, again, from the outer groove, this interventricular groove, to the inner groove of the heart. And you can see by cross-section, this really spectacular pattern of this band of red cells, where the red cells contribute to what will become the interventricular septum. So this stage is just at the beginning of as the heart forms the IVS. And the red cells occupy sort of a morphogenetic nexus, regions that are important for when we think about sort of the variation of ventricular septal defects. So it includes sort of occupying this region of the AV canal into the sort of the beginnings of the atrium and interatrial septum and the outflow tract. So we think that this location is really pretty striking in terms of we think that may correlate to have some importance for ventricular septal defects. Next, we wondered, so here we went in detail. We looked at how these cells sort of progress during development, during heart development. And we wondered, are these cells important for anything? Are they essential? And so here what we did is we misexpressed diphtheria toxin. So wherever diphtheria toxin is expressed, it will kill the cells. And we used this sort of intersectional sort of reporter. We modified it, so we knocked in the diphtheria toxin into a construct, into another safe harbor locus, so that we could then look at the lineages, as well as sort of ablate these cells of interest. So here in control, we're looking at a mouse embryo. Here's the developing mouse heart just after looping. You have the left ventricle and the right ventricle. Here's the outflow tract. We're looking at it from the right side. In mutants, we observe RV hypoplasia, which is apparent grossly. And we see this sort of pattern again at 12.5. Here's a wild type heart, left ventricle, right ventricle. Here you can see this sort of lumpy, bumpy appearance of the RV, as well as this RV hypoplasia. And you can see some red cells here, these double positive red cells. And in the diphtheria toxin mutants, we see that there's sort of disorganization of the interventricular septum. It's not as compact here as in here. And then again, you can see this sort of lumpy, bumpy appearance of this RV hypoplasia. We think that there might be some LV hypoplasia, but it's certainly more striking on the right side. So this really tells us that this sort of lineage that contributes to the interventricular septum, it not only is important for the formation or development of the interventricular septum, but there's also some signaling that's happening that is interacting with chamber formation. So, which is a bit of a surprise that it's also affecting chamber development. So next, we wondered if we talked about those lineages earlier about the right side and the left side. So we wondered if by ablating these cells, what would happen to this sort of lineage boundary that I showed earlier. And there's a concept of the lineage boundary, which is this boundary where they have a sharp boundary with the lineage. And then there's a specialized type of lineage boundary is known as a compartment boundary. And a compartment boundary is a boundary that's actively managed to segregate really two different fields of cells that have different functions. And the goal really is to prevent the mixing of these two cells. So in compartment boundaries were first sort of identified in fly and fly wing in imaginal discs. And there are really less than a handful of examples of the compartment boundary that have been reported or discovered in the context of mammalian development. So we wondered if this might be another example. So here I'm showing you sort of a representative light sheet sort of optical section. I'm in the left of the green lineage. This is the TBX5 lineage. In the sort of magenta is the MEF2C AHF lineage. And what we did here is we looked at different levels. So here in this dashed line represents sort of an example of a linear profile across from the right ventricle to the left ventricle. And we looked at sort of several levels. And so, for example, here is sort of the manifestation of that, looking at just the TBX5 lineage. So we see that in black represents wild type, green represents mutant. So in the black line, you can see that the by linear profiles, when we quantify sort of the intensity, sorry about that, that we have an increase in the TBX5 lineage on the left side of the heart. This is kind of represents the left side of the heart, and this represents the right side of the heart. Likewise, in the MEF2C AHF lineage, we see that the MEF2C AHF lineage is reduced on the left side, but increased on the right side. When we look at sort of the DTA mutants, so here in the green, what we see is we see a shift, a leftward shift. So from this peak to this peak, which I've represented here in this green arrow. And then we also see so it's leftward on the screen, but it's actually towards the right ventricle. So it's actually a rightward shift of the green cells. So here's an example of the green cells that normally almost never are in the right side of the heart, but there are cells with the ablation after DTA, that there are green cells that kind of spill over into the right side of the heart. And as well, the location of the magenta cells, they are not as far left. They're also sort of shifted to the right, but they're not shifted sort of in synchronization. They're actually the TBX5 lineage shifts more to the right, and the MEF2C-HF lineage also shifts to the right, but not to the same degree. And so this kind of represents lineage mixing. So it's this idea that basically these cells are not segregating when you ablate these cells, and there's mixing. So we think this is really good evidence that these double positive cells are marking a compartment boundary in the interventricular septum, and that they're required for this compartment boundary. Okay, so next we wondered what sort of genes might regulate this. And so since TBX5, as I mentioned, is earlier in the talk, TBX5 is linked to congenital heart defects in the context of Holt-Oram syndrome. And so because TBX5 marks these cells early on, we wondered if reducing the levels of TBX5 might affect this lineage, at least later in development. And so here, what I'm showing you are two hearts. So this is a sort of a control heart, and this is a mutant heart. Similar to what I've shown you before, you see these sort of red cells that are really nicely aligned. They're stacked like coins, basically, near the apex all the way up. In the sort of mutants where we reduce the levels of TBX5 in the green lineage, what we observe is we see ventricular septal defects, which are shown here in this asterisk. So you can see the sort of communication of the blood between the LV and the RV. And then the interventricular septum, there are fewer red and green cells, and these cells are not stacked like coins. They're kind of disorganized. And as well, that their sort of pattern of where they are in the center is also a little bit broader. So we quantified this. So first, this is a sort of a representation of how we quantified the orientation, taking it again from sort of the fly wing, how fly wing hairs are organized, and you can see the sort of linear trajectories of each of the cells, and that this is mixed in the context of the TBX5 mutant. Next, we looked at sort of the spread of the lineage. What we observed in the TBX5 mutants is that the position of the red lineage is shifted over to the left side, so the boundary shifts to the left, and also that there is mixing, so that there's a broadening of these sort of red cells, so that the boundary is not intact. So there are two things happening here: that there's a change in the boundary position of where the cells are, as well as the integrity is also compromised. And those distinctions will be important later in the talk. Okay, so we wondered what might be downstream of TBX5. So here what we did is we undertook a single-cell RNA-seq experiment to identify sort of potential downstream genetic regulators of TBX5. So here we microdissected hearts at embryonic day 13.5, so just before the finish of ventricular septation. And so we isolated different parts. We isolated the left ventricle, the interventricular septum, and the right ventricle. We did that both in controls and mutants. And so we did a series of analyses. I'm just going to cut to the chase. So for those that don't know, single-cell RNA-seq is basically single-cell RNA sequencing of each individual cell, so we can get basically the gene expression of each individual cell. So this is what's called a UMAP. So this is a sort of a representation of where each dot represents a cell. And the relative sort of locations of one dot to the other happens to, it really reflects the relationships of one cell versus an adjacent cell in the context of this sort of reductionist space. So that's just to say it's to identify how cells are related. So here what I'm showing you is this UMAP that represents the two sort of genotypes. And then this UMAP represents the different sorts of cell clusters that we observe. And through a series of analyses, I'm just showing you one here, we noticed that there was one cluster that was predominantly control, this cluster 5, and this other cluster that was predominantly mutant. And so we wonder what was the difference between these sort of controls and mutants. And what we observed is that in the mutant cluster, we saw that there was a reduction in genes that are expressed in the AV sort of AV canal, AV valve region. And in the also in the mutants, and so that's shown here. And then, so this is a dot plot just showing the sort of relative amounts of gene expression between the two clusters. And in the mutants, we also saw that there was an upregulation of trabecular genes. So trabecular genes are generally in the trabecular and the RV and the LV, generally not in the interventricular septum. And so what we saw were a group of genes, some sort of classic trabecular genes that were upregulated in the interventricular septum. We also noticed these two genes, which are known for axonal and vascular guidance and patterning, and hadn't really been described in heart development. So these really kind of caught our eye. So what we did is we confirmed these results using a different sort of experiment. So here we're using basically fluorescent in situ hybridization of RNA. So SLT2 is expressed in the trabecular region. So here, here's the heart, here's the LV, the RV. You can see it's expressed really in the trabecular region. It lines kind of the borders of the interventricular septum, but not the core. And this is a blow up here of the interventricular septum. In the TBX5 mutant, where TBX5 is reduced, what we see is that that SLT2 kind of spills over into the interventricular septum. So it spills over into the interventricular septum, and interestingly, it also spills into the compact layer. So for example, here, SLT2 is absent in the compact layer, but in the mutant, it's spills over into the compact layer. And it's thought that the interventricular septum is in many ways compact layer-like. So that's all kind of consistent. SLT2 is expressed both in the LV and the RV. Netrin-1, on the other hand, is really predominantly expressed in the LV, in the sort of trabecular layer. And it also has this pattern in the interventricular septum where it has a gradient in the interventricular septum. It's highest on the left side of the IVS, so over here, and then it kind of gets reduced to the right side of the IVS. In the TBX5 mutant, like SLT2, the netrin sort of spills over into the compact layer, and this gradient gets changed. There's no longer a gradient. It's blunted. It's reduced, but it's blunted. So the pattern in the context of the TBX5 mutant is reduced. So we think that that what might be happening is that with the reduction of TBX5, you have these genes that should be in the IVS, or that they're normally in the IVS, that their expression gets dysregulated, and perhaps this might cause some of the defects that we're seeing in the TBX5 mutant. So next we wondered, well, what's the role of SLT2 or netrin-1 on their own? And so here we generated knockouts of SLT2 and netrin-1. So here I'm showing you some data from of SLT2. So here's a sort of a wild type comparison of the LV, the RV. Here's the interventricular septum. In some hets, we observe a sort of a muscular VSD, a communication between the left ventricle and the right ventricle. In homozygous animals, we saw an increased incidence of, now we're seeing both membranous VSDs and muscular VSDs. And we also saw this sort of hypertrabeculated interventricular septum. which is it's a relatively rare. It's not too common in the context of looking at sort of ventricular septal defects in mice to have this sort of hypertrabeculated IVS. But nevertheless, we see that here in the SLT2. And what's striking is that we also see a sort of overlapping phenotype with the Netrin-1 mutants. So we see membranous VSD in the het at low incidence, and then we also see both muscular and membranous VSDs. I'm showing you a membranous VSD here, as well as this sort of hypertrabeculated IVS phenotype. This is interesting because Netrin and SLT, sometimes they work together, sometimes they work, opposing. So this would really kind of suggest that potentially that Netrin and SLT2 are working cooperatively here. At least that's the hypothesis, to help with interventricular septum, given that there is overlapping phenotypes. So next we wondered, well, what is the role of Netrin and SLT and Netrin in the context of this lineage? And so here we did sort of a similar approach where we looked at this double positive lineage. So this is in magenta. This is that double positive lineage that sort of lines the interventricular septum. It does have some cells that spill over normally into the LV, but for the most part, it's in the interventricular septum. And so here we're checking linear profiles at the base, middle, and near the apex of the heart. And we did it at a couple of different levels, both at a sort of anterior, middle, and then posterior. And so here, for example, here's a Z-stack projection of the lineage in the context of a mutant, a representative mutant. And you can see that this sort of red is much more spread out than in the control. And when we quantify this by sort of the linear profiles, black again is wild type, and the sort of magenta is the SLT2 mutant. You can see that the lineage is still centered on the IVS, but it's a little bit broader. And what that means is that the broadening pattern means that this sort of boundary that I brought up earlier, that there's mixing happening. So really the boundary integrity is not maintained. So this is somewhat consistent with a part of the TBX5 phenotype. So we looked at Netrin. So here's a wild type littermate control of this Netrin mutant. So here the picture is a little bit different. So in the SLT2 mutant, we saw sort of the boundary is more diffuse, so that means that there's lineage mixing. Here in the Netrin-1 mutant, you can see this kind of in the linear profile. What we see is that the position of the boundary shifts to the left. So the boundary is intact, but it shifts to the left. And you can see that here where you have this peak shifting over to the left. And it's and so in many ways, this is sort of complementary to the SLT2 phenotype, and together the Netrin and the SLT2 phenotypes recapitulate the sort of the lineage phenotype in TBX5. So to summarize this part, what we've shown is that we think TBX5 regulates this lineage, and the role of TBX5 is to organize this boundary so that the left ventricular sort of lineage cells and the right ventricular lineage cells, they stay segregated. And TBX5 does this in a way that sort of regulates both Netrin-1 and SLT2. And I didn't have time to show you, but we have evidence that that TBX5 likely directly regulates Netrin-1 and SLT2 because chromatin occupancy of TBX5 is at the promoters of both Netrin and SLT. And then in the context of reducing the levels of TBX5, you kind of take off this break and you have a misexpression of SLT2 into the interventricular septum, and then you have this sort of abnormal expression of Netrin-1, and this sort of leads to this sort of mixing that both sort of interrupts boundary positioning and boundary integrity. So I've highlighted, so, this work is done by lots of people. I'd like to especially thank my mentor, Benoit Bernot. And then through the talk, I've sort of mentioned different people who have contributed to different aspects of the work. I'm happy to start to take questions now. Yeah, thank you all for your attention.
Thank you, Irfan. That was really, really interesting stuff. Just while people are giving people a chance to come on, I was interested in the, and maybe the cardiology colleagues on here can help a little bit, but the pictures of the netin and SLT2 cats and homozygote mutants, like with all that trabeculation, does that kind of look like a non-compaction? Is that related to ventricular non-compaction, you think? Or, and do people with non-compaction have increased rates of VSDs?
So as you know, I'm not a cardiologist, and there are a couple of cardiologists, at least fellows that are on, but I'm not going to put them on the spot. So I don't know the answer to that. My sort of, generally, I think that they're not necessarily associated, but I could certainly be wrong. What we see is kind of interesting is that the sorry, I have the Zoom window, is that the compact layer is also thin. So there's something complicated going on. I think what's happening is that there's this balance between the specification of the compact layer and the trabecular layer. And I don't know if it's basically there's that because with the trabecular layer, becomes more compacted during development. And typically that's apparent at 14.5, which is where this time point happens. So we're trying to figure out whether or not how that balance might work. But also with non-compaction, I don't know, I've taken care of patients with non-compaction. This sort of hypertrabeculated IVS, it doesn't really, it's not reminiscent of that, but I'll defer to the other experts
no,
in the
certainly
room.
not at the interventricular septum. I know Ron is on. He can be put on the spot. We won't put
Oh, good.
on the spot
Ron
to know.
Yeah. I would think of it a little bit differently with regard to, I mean, first of all, we probably don't know exactly what non-compaction is and whether it's just a variant of some other kind of cardiomyopathy that we see. But I agree that I wouldn't really necessarily say that's related to VSDs or what the TBX is, what you're proposing TBX is responsible for. But it doesn't really look like the pattern, and it doesn't really look like it would meet the criteria for non-compaction. But I think about the non-compaction as being more of a cardiomyopathy, and then you're kind of going down a different road. I guess the clinical question that I had here is, I'm trying to understand how this relates to the different VSDs, because we definitely have pathologic VSDs, but then the VSDs that, at the very beginning, you said that many patients have VSDs, and yes, we see them all the time in the newborn nursery, and then they go home and they resolve within, weeks to a month or so. And I've always kind of explained that off as just normal development that was just slow to complete, as opposed to what you were showing in one of your diagrams as a muscular VSD related to one of these mutant genes. And where's the break? Where does that break? When is it just normal development, and when is it related to one of these mutants, as far as how do you counsel a family with a kid with a VSD? Does the muscular VSD that closed at a month, does the subsequent pregnancy have an increased risk? or is that just slow development and have no relationship to this finding?
Yeah, I mean, that's what a wonderful question, right? Like how do you know, how do you distinguish what will be pathologic and what won't? And that's, I mean, that's just a terrific question that we kind of hope to eventually get at. Because as you know, many muscular VSDs, they kind of close on their own or they're not hemodynamically significant. So we're trying to, it's hard for us to figure out what is hemodynamically significant. Basically, what we use is, particularly in a mouse embryo, what we use is lethality. So these mice, they die early. So we think that there's something happening, and I think what's happening is heart failure. And given that there are ventricular septal defects, I think that's probably the cause. Most mice, when they have a ventricular septal defect, they don't last beyond the perinatal period. Generally, they die between day 3 and day 7. That's probably when their PVR drops. And my guess is that it just gets so exacerbated because their heart rate is, it's 300 to 500 beats per second. So they just enter into the cycle of overcirculating and having heart failure just a lot sooner. So that's why they die earlier. But we can't really do echo to look at gradients and that sort of thing. But that would be cool. But great question, Ron. Thank you.
Thank you. Does anyone else have any other thoughts or questions?
This is Yao. I have a basic question, Irfan.
Hi,
It sounds like what you're describing is really a physical phenomenon in terms of the boundaries and stuff, and that the cells, in terms of their expression, they continue to be differentiated just because of that physical location. I'm wondering whether there's any evidence that there may be either flow or pressure related phenomenon that helps maintain that boundary in the developing heart.
Yeah, that's a really great question. I mean, we don't have the answer yet. I can say that that the boundary that reflects this sort of lineage or the compartments, the boundary really nicely aligns very well with how the myocytes are organized between the left ventricle and the right ventricle. So there are studies where they've looked at sort of what's called a myofiber arrangement, and at the end of ventricular septation, at the same time points that I've been showing here, that's when it starts to become apparent that you can see with the interventricular septum, you can see that that part of the interventricular septum is, obviously it's the septal part of the LV, and so it's in alignment with the LV, and then the right side is also in alignment with the RV. So that doesn't necessarily answer your question about flow, but in many ways it does, because the anatomic arrangement is getting set up in anticipation of the flow and the changes that happen after birth. But it is really kind of interesting. I think it would be hard for us to test at the moment using current tools, but it's a cool idea. So thank you.
Okay, well, thank you so much for joining us, Irfan, and we'll see you all next in a couple of weeks.
Awesome. Great.
Bye,
Thanks so
everyone.
much.
Thanks, Irfan.
All right. Take care.